Good morning, afternoon, evening, wherever you are, and welcome. This is Freethought Hour, and wherever you are, up a tree in the Philippines, we don't care. Or if it's tomorrow, you know, you don't have to attend a live thing. This is the modern world. Scheduled viewing is pretty much yesterday, and now we all want to play on demand. So welcome. Anyway, tonight, it's tonight here where I am. I have a fantastic guest. This is Dr. Seth Garren, who is the lead of Pfizer's well, let him tell you himself. Seth, how the devil are you?
I'm good, thank you.
Excellent. So what is your actual job title?
So my job title is I'm the lead of the Sequencing Technology Center at Pfizer in Boston. So my responsibility is to basically identify new sequencing technologies for gene sequencing and evaluate them and see if they're appropriate for bringing on board for helping our scientists answer questions about disease.
I'm going to ask you more about that in a minute. But first of all, can we have a little bit of a nose into your background?
Sure. So
how did you get to be the Seth you are today? I'm going to full screen you and let you tell us without me being visible. Okay?
Sure. So I guess my background is I was raised Presbyterian, and I grew up in Connecticut, so eastern side of the U.S. And I was raised Presbyterian. My father was actually a minister, but my parents weren't super religious or anything. They were sort of nominally Christian with sort of very private beliefs. But I went to Sunday school and I sort of paid attention to that. And as a result, I developed the type of literalistic beliefs that a child would typically have up until about middle school when I lost my faith. And that was mostly surrounding my obsession with the afterlife and what
Can I just pick on something you said?
Yes.
Your father was a minister.
Yes.
But your family wasn't overly religious. I mean, how does that work? Can you get away with that in the, what did you say, the Pentecostals?
Presbyterian.
Presbyterian. Yeah, you certainly wouldn't get away with it in the Pentecostals, would you?
Well, so the answer is it depends. There's a variety of denominational conservatism that you can find across religions. Today, it tends to be a bit more conservative as the more liberal mainlines are kind of a dying breed, and they kind of become less defined. But back in the day, you would have a broader variety of religiosity, especially across my parents' generation.
That's a trend, isn't it? We've gone from being a nuanced culture into a polarized one in quite a lot of respects.
Yes, definitely. So with my parents, I think they grew up in an environment where everyone was at least nominally Christian. Like you'd ask them, do you believe in God? They'd say yes. Do you believe in heaven and hell? And they'd probably think of it like an afterthought, like your almost cartoony vision of it. Oh yeah, heaven's got clouds, angels, harps, halos. Hell's got devils, pitchforks, fire, etc. And they wouldn't think more than that. And just everyone was religious. Nobody questioned it. You just went to church on Sunday. It's just what people did. It's a form of eating your vegetables, so to speak. And I guess they came from a background where they didn't think about it that much. So this is different from your more hardcore fundamentalist guests, where they come from the background where the religion becomes the center of everything. Despite the fact that my father was a minister and the religious ceremony and ritual was the center of things, at least from a logistical standpoint, the belief wasn't the center. And so they would focus more on the ritual, the history, the feeling, the communal interactions.
Do you think he was a closet non-believer?
Oh, I know for a fact he was a closet non-believer, as well
Really?
as he was also came out as gay when I was in college. So,
yes.
So there's a number of things that were not in line with what some of the more conservative congregation would have expected.
Definitely not a stereotypical preacher there.
Correct. So that's but as a result, though, because people knew I was a son of a pastor, there was a number of expectations where they'd assume I was religious, or at least under the auspices of religious oppression.
But you seem to have managed to escape, because, you know, if this had happened more recently, we would be recommending your father. I don't know whether he's still around. I haven't asked you that yet. We would probably be sending him off to, what are they called?
The Clergy Project.
That's the ones. Yes. Yes.
Fortunately, he's wasn't in the Bible Belt per se, though he did have a couple of, I like to call it a sheep and wolves clothing situations, especially in the 90s. But eventually he ended up in a very accepting and liberal and fairly wealthy but tiny church. And then he ended up retiring a few years ago. And now he lives in Canada with his husband. And he actually met someone who had almost the exact same life story as him there. A guy who had 2 sons, was a pastor, came out as gay. So I guess there's some similar travelers that kind of arrived at the same location.
Yeah. And Canada, of course, is very liberal too, much more so than the Bible Belt.
Yes, absolutely. Although we never went that far. Although I will say one time, I was born in Southern California, and in 1990, I was born in 84. In 1990, we moved to Connecticut. And during that time, my dad was looking for new jobs, and he was kind of throwing his resume out as you do. And he had one job interview in like the sort of Northwest, like Washington or Oregon area. And he went to the church, and they asked him, just stone cold, absolutely serious, like, what is your experience with demonic warfare? How do you deal with demons? Because we have a problem. We've had some spotted on the roof of like the school, and we need to have someone who has the proficient, like, and it's like something out of like, you know, a science fiction movie or something, you know, and he's just like, I'd never even heard that question before.
They were sounding him out for possible exorcisms.
Apparently. So, I mean, that was one of the more exotic job interviews he's been on. But I mean, I guess when you work in religion, there's all sorts of expectations and premises that you may forget other people have.
So you escaped being indoctrinated. That was good. And then when you were in your teenage, you turned properly atheist. What happened to do that? What was the trigger?
When I was in my early teens, I was very sort of fixated on like death and mortality, because just the concept kind of sinks home when you become old enough to understand it, and you have to come up. People have different responses. Some just sort of shove it in a drawer, don't think about it. Others just say, "Oh, you go to heaven and that's that." But I was like, "Do you really?" I don't feel confident enough that that's actually the case, and so I was very fixated on it. And I remember hearing about atheism, and I thought like, wow, it would take somebody who's really brave to just bite that bullet and just say, no, you actually like just die when you die. And so I was very preoccupied with it. And I think it was that preoccupation that was driving most of my incentive to believe, because otherwise it's just, you know, most kids my age would just be dragged to church. They wouldn't pay attention and they wouldn't think about it. But I was just sort of very fixated on that aspect of it. I also was somewhat nervous, because I took literally the idea that there is some being with all power and kind of inscrutable, like, you know, motives, judging every action I do. So if I would have an errant thought, I would start to worry if like a stray thought would get me in trouble, and that would sort of torment me a bit.
Yes, it's the God as closed circuit television in the sky, isn't it?
Precisely, yes. As Hitchens puts it, the celestial North Korea. And ironically, what I tell people is that the kind of moment where I lost my faith is ironically when I saw the movie What Dreams May Come, which for those who aren't familiar, it's a movie starring Robin Williams, came out in the late 90s. And it follows the story using a fairly modern Catholic interpretation of the afterlife, whereby, you know, him and his children are killed in a car accident, and he's in the afterlife, and then his wife is left behind, and she gets severely depressed because her whole family died, and she eventually commits suicide. And per Catholic tradition, suicide is an unforgivable sin that sends her to hell. So then he goes on this journey to try to, like, you know, leave behind paradise and rescue her from hell. And so it has a lot of this Paradise Lost style imagery. And the particulars aren't important, but I think what really sort of hit home for me is that like actually, like people, very few people are willing to like describe the afterlife. They like to say they believe in it. They like to speak of it in vague terms, like, oh, spending eternity with God or seeing your loved ones. But they never talk about like the minutia, the subtle world building of it. And seeing a movie that attempted to, I think, really hit home how just completely hard to believe this concept is. Because when you get into like, what do you look like? What kind of body do you have? How do you eat? How do you do any of these things? And just seeing the subtle minutia, the fact that, oh, anybody could just look like anyone else. It just occurred to me that it's like, that's the moment I just lost all ability to sustain credulity. Yeah, this is all just wishful thinking.
I've not seen that movie, but I'm going to put it on my to watch list.
It's a beautiful movie. It's extremely vivid. Likewise, because it's the only time I've seen anyone attempt to, explain what that looks like. But,
and of course, if you're going to do a movie about the afterlife, you have to have sets. You have to have backgrounds. You have to have some physical representation.
Yes, and I know, like, you can easily judge that. Well, obviously, it's not literally like that, but I think just the fact that the moment you think about the non-obvious mundane elements of something, which I think are the foundation of what makes something feel real, that's what killed my ability to believe it. And I think a lot of people seem to avoid that. Obviously, there's not much to go off of. The Bible doesn't say like, oh, heaven has these precise parameters. So it's pure speculation. But any form of speculation you make grounds it into something that doesn't make sense.
Nobody's come back to tell us what it's really like, so we can only speculate.
Well, with the exception of the occasional like 8-year-old who drowned for 4 minutes and wrote a book. Like,
well, they didn't die.
Yes, did they?
They made a lot of money, and under the direction of his father, I think, in that case.
Yes, that was my understanding as well.
Yeah. So you've got to the stage where you've seen the Robin Williams movie, and at that point you decided to style yourself an agnostic.
Yeah, I think at that point I lost the ability to believe there's an afterlife. So I let that go, and I'm like, okay, I'm reasonably certain that when you die, it's no different than before you're born. And I just kind of had to come to terms with that, because I could no longer sustain the afterlife belief. And at that point, there's no incentive to care about what God wants anymore, aside from him maybe like physically attacking you for some reason. There's nothing left to motivate it, which brings us back to our pre-show discussion about like why everything has to happen after you die to be the actual test. And then at that point, I became like, without the motivation to sustain the belief, I'm now taking a more unbiased approach to it. And I'm like, well, I don't actually know if there's a good reason to believe God exists or not. And here I'm like in high school. So that was sort of as much as I understood about it. And then it wasn't until college when, the new atheists started coming out. You have post 9/11 and you start seeing all these writings on it. And I'd see things like the God delusion. And to me, that seemed taboo. Like atheists are out there, but the idea of like questioning religion seemed just incredibly obnoxious and inappropriate and rude. And like, it just seemed very ballsy for someone to publish an entire book just saying what a lot of people thought, but just didn't want to offend anyone about.
Well, it would seem ballsy in America because the US is such a religious country. It's an outlier in the Western world, but Dawkins is an Englishman. He didn't
Hitchens.
Yes. They didn't think in American terms about what they were doing. It didn't seem to be that outrageous and courageous to them, probably.
And I think it took somebody with a more permissive culture to do it. I don't think somebody from Saudi Arabia is going to be the first one to publish on problems with theism.
No. Interesting.
So my exposure to that sort of made me think more about what I identified as. And I think Dawkins just flat out said, like, look, this isn't about having absolute certainty about anything. It's about saying, like, is there sufficient grounds to say this is the case? And there isn't. So, it's okay to say you're an atheist, you don't believe. And I'm like, okay, I feel more comfortable just saying, I guess I'm an atheist.
Yeah, that's right. My argument is we're all born atheists. After all, when we're born, we can't speak. So nobody can have taught us any religion.
I mean, I get a little confused with some of those like our rocks are babies atheists and lack theism and burden of proof shifting stuff. I guess technically on that definition, you're correct. But then it gets fuzzy as to what someone else means when they use the term. And so with my college experience, I had this one moment where I was on campus and there was a campus preacher. And the preacher started like, he was one of those, stereotypical fire brimstone types. And like, I started debating with him about that stuff, saying like, well, how do you know this? And what is your rationale for that? And pointing out issues with his examples of why he thinks this is reliable. And it kind of drew a little bit of a crowd. And so afterwards, a couple of students approached me and they said, yeah, that guy seemed a bit extreme, but I like the way you articulated yourself. Full disclosure, I'm from the campus Bible group, but we thought it might be interesting if you joined us and just had a conversation. And I thought,
this happened outside in the campus somewhere, did it?
Yeah. Just in the little campus walkway randomly. And so I said, you know what? Yes, this sounds interesting. Like this is a new opportunity to get exposure to some drastically different points of view, because I've never actually sat down with someone and had a conversation about beliefs, knowing ahead of time that we had diametrically opposed beliefs.
Did you know that what you'd just done would now be called street epistemology?
Well, it's kind of the reverse of it, I guess, because I guess street epistemology would be if I stood there and I'd ask random people, like, what's a belief you have? Let's figure out how justified it really is. But I suppose it's the equivalent. I think I've seen Magna Bosco do that to street preachers. So you're right. That would technically count. And so then I joined this Bible group, and my first time there was great. There was this older gentleman who was like a retired engineering professor who was just sort of leading the Bible discussion. And then afterwards I met with him and had a conversation. I said, I'm an atheist, but I was invited here and I'm just kind of interested in learning more about like why people believe this particular interpretation. And I was talking to him and he said something that kind of threw me off. He said, I wouldn't worry too much about the specifics of the belief. I think it's just important to have a good because I said so in order to point the students in the right direction, which is absolutely the last thing I expected. I wasn't sure if he was telling me something because it's something an atheist would be receptive to, or is this guy literally like manipulating Christian college students to behave by using their own beliefs that he doesn't even subscribe to? So either way, that was interesting.
And a deception too.
Yes, which is not, I feel icky if I'm going to lie about what I believe, but I understand that there's certain strategies that can successfully manipulate someone if you work within their belief structure.
This is how manipulation gets a bad name.
I suppose. I mean, what's interesting is that the second time I went, it's fortunate that wasn't the first time I went, because the second time I went, there was this one guy who kind of drank a little too much Kool-Aid, and he just went full on into like all the grosser parts of religion. And like I came into the event and it was like dark and they had like a PowerPoint presentation or something. So I came in and sit down and he's showing all these like pictures of starving children in impoverished or war-torn countries. And I'm like, okay, it's probably gonna be like Christian outreach stuff. No, it's not at all what he was talking about. He was saying, he's going on this long litany of how these children are suffering and how like awful and horrible it is. But instead of saying like, here's some Christian outreach efforts to solve it, which is what you'd expect, he's saying, but as much as they're suffering now, it's nothing compared to what they'll suffer when they die, because unfortunately they haven't accepted Jesus as their savior. And, we just have to mourn their lack of access to that, and this should be a cautionary tale for why, you should accept Jesus as your Lord and Savior. And we just have to pray that some Christians get to them before they die. And I'm like, wow, if I were an atheist trying to like smear the religious faith and make it look like untenable and disgusting. I could not have done a better job than that person who was ostensibly a genuine believer. And I was talking to the other Christians, like some of the other students afterwards, and they were like, "Yeah, that was a bit much." Like,
they say that the Bible is the best recruiting agent
for atheism.
Yeah. Well, anyway, I think you've explained how you got to be the atheist you now consider yourself to be. So, can we talk a bit about your work, because that sounds amazingly interesting, or at least it does to me as an ex-science teacher.
So, were you interested in my current work or my like kind of academic journey?
Well, both really, because you did some research into pancreatic cancer treatment.
Yes, one of my things involved that.
Yeah, because pancreatic cancer it's almost a death sentence, isn't it? There's a small number of people who survive it these days, but it's, in the low percentages, isn't it?
Yes, and the reason is that it doesn't really show symptoms until after it's too late. So it's not that pancreatic cancer per se is more lethal than any other cancer. It's just it's so buried inside of your viscera that like by the time you know anything's wrong, it's too late. So along those lines, there has been a lot of interest in coming up with less invasive methods of interrogating. So you can't just like have massive thoracic surgery on the off chance you have something to just start taking random solid biopsies. So as a solution to that, the community has been looking heavily towards what's called liquid biopsies. So the idea is that the tumor is going to secrete a whole bunch of molecules and vesicles and objects into the bloodstream that would not normally be present in a healthy person. And so we were looking for different categories of biomarkers, if you will. The typical biomarker would be something like a protein or a metabolite. So when your body like eats something, so for your audience who's not familiar with that, metabolism is just like your body eating a thing, right? So if a cell eats something, it'll like take a molecule of like sugar, break it into smaller pieces, and then get the energy out of it. And the smaller pieces are left over. Those are called metabolites. And so there might be specific metabolites that are small pieces that could float around in your blood that are disproportionately associated with cancer. You could also have little vesicles, which are these little balls of lipid membrane that are like mailing packages that cells can release, and they'll send signals around the body. And you can also have nucleic acids. You can have RNAs or cell-free DNA that float around in the blood. And sequencing technology had just gotten to the point where it was sensitive enough to detect that reliably. And so there were a whole bunch of academics and startups trying to test, like, could we take blood from these cancer patients? And now that we know who is and isn't a cancer patient after the fact, we can take their old blood samples and compare them. So
what's interesting there, because metabolites are going around in the blood all the time, but they're normal ones. What you were trying to look for was ones which were only occurring in the presence of a cancer.
Yes. So that's a part of the entire liquid biopsy paradigm, is to say, are there any objects in the blood that are unique to cancer that show up before the tumor is problematic or past that point of no return?
Well, I can intersperse a little story here, personal story, because my first wife had one of these not detectable until it's too late cancers. It wasn't pancreatic. In her case, it was ovarian, which of course has the same problem because it's deep inside the abdomen, and it can't be detected until it's developed to a condition where it's virtually untreatable. There are a few women who can be operated on, but there's not many. It has to be chemotherapy, really. So
that's a good illustration of the unfortunate challenge that faces us. But it's also a good like segue into the second half of that work that I was there. So this is when I was doing my postdoc at Mass General Hospital. And so my first year I was doing liquid biopsies, but my second year I was actually studying what's basically our most cutting edge cancer treatment, which is called immune checkpoint blockade. And this is the thing that has moved the needle the most in the last 10 years. And now everyone's kind of trying to find ways to augment this effect. And the way it works is typically you have tumors pop up in your body all the time, and your immune system just takes them out, and nobody's the wiser. You don't have to get any doctors or chemo involved. And so
just like in a smaller way, we have warts appear on our body, and then subsequently they go away.
Or other endemic viruses. There's viruses that live inside of us that you don't see unless you get HIV advanced stages. So there's a lot of things that we take for granted with how our immune system works. Well, so naturally, when you have cancer to the point where you're talking to an oncologist, that means that it has somehow evaded your immune system.
Yes.
And so there's this entire field of research called immune oncology, which specifically specializes in the interface of the immune system in cancer. So it's not merely what chemicals can we throw at it. It's more, how can we jumpstart the immune system to attack the tumor? Well, one of the most successful interventions that I think has, in a couple of cases, has increased survivability rate by like 30%, which is huge, has been what's called checkpoint blockade. So what happens is the immune system has these things called checkpoints. So they have these surface receptors on the cell that look to see whether or not the cell they're attacking is a self cell or a foreign cell. And so the self cell has this protein called PD-L1, and it expresses on the surface. And if the immune cell has a complementary receptor called PD-1 detects that, it will then say, oh, I shouldn't attack this. This is a self cell, and that's a checkpoint it has to go past, which is saying check to see if there's PD-L1. If so, stop attacking. So what'll happen is a lot of tumors will just flood their surface with this "don't shoot, I'm innocent" signal. And it's the equivalent of like the mob boss saying to the police knocking on his door, "Do you have a warrant?" And they're like, "No, we don't have a warrant," and they walk away all grumbly. Well, immune checkpoint is, let's take an antibody that binds to that PD-1 receptor on the immune cell so that he just doesn't care anymore. And so he says, I don't care about your warrant, and just kicks the door in anyway and just attacks him anyway. And that's called immune checkpoint blockade. And so
how do the tumors present self-identity?
They basically, the tumors that happen to be expressing more of that PD-L1 gene, the don't shoot signal, are the only ones left. So the immune system comes in, they kill all the tumors that don't have it, the ones that have high levels of it, and it's recovering. And then they're scrambling their DNA so that the ones that happen to get even higher levels start to survive. And then what happens is the immune cells come in, they get all of this. Do you have a warrant? 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 And they just decide, I'm done, I'm quitting. And they shut down and they become exhausted. And so you end up seeing a tumor that's full of immune cells that have just given up.
So that's why in the old method of blunt chemotherapy treatment, it kept coming back until it wouldn't respond to the final attempt at chemotherapy. And this is interesting because it's a sort of internal and condensed example of natural selection.
I think it's the perfect example of natural selection, and I refer to it frequently when I encounter creationists or intelligent design proponents who like to poo-poo the random selection process without realizing that that's primarily what drives carcinogenesis. Like, we call something a carcinogen because it damages DNA. And it's the process of damaging DNA that we also call a mutator phenotype. So if you have a particular type of style of cancer whereby the cells, like DNA repair genes, are all broken, then it accumulates mutations like crazy. And that can drastically increase the variability. Now, of course, a bunch of them will probably die from their own, broken gene expression, but it also it's like this gauntlet that they put themselves through, and then you get more dice rolls, so to speak.
Well, I want to take you on a slight rabbit hole here, because this is a sort of reverse example of autoimmune diseases, possibly.
Yes.
Where
So that's also a brilliant segue, because that's what my current department specializes in. So I went from both ends of the spectrum. So during my postdoc, I worked on immune oncology. And then when I came to Pfizer, the department that I sit in specializes in autoimmune disease. It's literally inflammation immunology. And so we're doing the same kind of thing, just in the opposite direction. The immune system has a variety of redundant and complex regulatory layers, as any biological system does. And when you're trying to do a therapeutic, you're trying to perturb that system in a direction that's more towards what you want. So if the immune system is sitting there not killing the tumor, you're trying to push it towards killing the tumor by increasing inflammation. But if the immune system is causing an autoimmune disease, you're instead trying to push it towards the anti-inflammatory regulation and decrease inflammation.
Yes. Interesting. And how ready is this for rolling out to treat people with both autoimmune conditions and cancers that have hitherto not been treatable?
Well, we have like Pfizer in particular has an R&D department in San Diego that specifically does immune oncology, and our Boston unit, Cambridge specifically, does autoimmunity, and we talk to each other a lot on those respective platforms. The immune oncology, like I said, uses the checkpoint blockade as their starting point, and then they try to come with ways to enhance it, make it better. Why does it only work in 30%? Can we move the needle up to 50 or 70 or whatever? With the autoimmunity, we look at what are all the signaling pathways that we see that are uniquely overexpressed in autoimmune cases compared to healthy or compared to successful treatments, and how can we then find drug targets for that. So, for example, Pfizer has a drug on the market called Xeljanz, which is a JAK inhibitor. So it inhibits one of the proteins whose job it is to carry a signal to increase inflammation. And there's a whole bunch of proteins that kind of create this Rube Goldberg daisy chain called the signal transduction cascade, where protein A turns on protein B, turns on protein C, and eventually...
You're talking about complement?
Complement is an example of a cascade. This is so the complement is creating a cascade to create basically a weapon. This is creating a cascade to create a signal. So it's the difference between firing a gun and calling 911.
Okay, yeah.
So in a sense, we're getting in the way of calling 911. So if Barbecue Becky wants to call 911 because she saw some Black people doing barbecue in a park, which is clearly an autoimmune example, we just prevent her from having a clear signal to the dispatcher. Whereas in the case of, if they're showing up and firing and you wanted to interfere with complement, you would jam their gun. And that's complement is one of the guns they would use.
Interesting analogies. The other little, before you carry on with that, the other little rabbit hole I wanted to take you down was sniffer dogs. Now, we're training dogs to detect cancer, and what they are doing presumably is identifying some of the more aromatic versions of these metabolites that are specific to a tumor.
Yeah, so it's the same principle. So instead of trying to get the metabolite out of the blood and put it into a mass spec, you're saying, well, nature's already created an extremely effective mass spec in the form of the dog's olfactory bulb. So if we can find a particular scent that we can correlate to the tumor, dogs are masters of correlating scents. They don't even need to know why. They just need to know if they correlate it, they get the treat. And so you have this correlating machine ready to go. And all you need to do is find it. So on the off chance there's a metabolite that can be smelled and can be detected by a dog and done so early enough, then that's just as good as a liquid biopsy win. So that's actually a brilliant example. Yeah.
It might be quicker or cheaper or something as well.
Another option is rats. Rats have excellent smell.
Yes. Well, there's a story about a rat that was trained to identify landmines, isn't there?
Yes.
And he died recently. He was a rather large rat, but he died with honor, I think, because of his track record.
But his advantage is that he's too light to trigger the landmine.
Yes, yes.
Now I don't know that cancer patients want rats crawling all over them, but they're probably more amenable to dogs, hence the rationale of that usage. But it's a very brilliant idea.
Yes, definitely. Yeah. So where to next? What's the prospect? What's the direction of your research in future?
So my actual job is now more technology focused. So I am embedded in the autoimmune group, but I also help support all of the groups at our site. So our site encompasses autoimmunity, rare genetic disorders, and metabolic diseases. And so like our technology is all based around gene sequencing. So we have all the latest and greatest Illumina sequencers. And then we also look for things like single cell sequencing. So instead of just taking your sample and normally you would like take a bunch of cells and you, for lack of a better word, put them in a blender and just take all the, the nucleic acids out.
To get your whole individual genome.
I mean, did you ever, when you were teaching, did you ever do the little like DNA out of an onion activity?
Yes.
So it's literally the same thing we do. Without just, we don't extract an entire onion's worth, but it's the same principle.
Did you get the little, was it a purple line that indicates where the DNA is?
Well, that's trisol. That's a bit harsher. I hope you're not doing that with students when you were, I mean.
Well, back in the day, we got away with a lot more because HSC hadn't been invented.
So you had your students working with trisol? Like
I can't remember, to be honest.
Trisol is pretty intense. So for those who don't know, trisol is it's basically a phenylchloroform mixture with guanidine thiocyanate in it. So it's highly, highly toxic and corrosive. But what it does is if you dissolve cells in it, it will rip all the polar and the non-polar parts of it apart. So all your lipids and your proteins will go one place, and then all your nucleic acids go to another. So it's a convenient way to separate them out. And so then you just add chloroform to it, and then you get this aqueous phase. So you just pull the clear liquid out, basically, and that has only DNA in it, and then you can then wash it off, and you get pure DNA. It's a nastier version of the high school experiment where you just stick the little glass rod in and pull it out, over the counter things. But it's faster, and it's just one of the methods. So then
don't try this at home, people.
You can do the onion one at home though. You can, if you just Google onion DNA, you can definitely do that at home. I would highly recommend it if you want to like teach your kids like how to pull all the DNA out of an onion or a strawberry. You can totally do that. Do not order trisol or phenylchloroform.
No. Not that you'd get it. You wouldn't have it delivered anyway.
You'd be surprised.
Oh well, it depends. Amazon today, who knows?
Yeah.
So you're trying to correlate the information that you get from gene sequencing with the information that you can get from microscopy. Now that's interesting.
So that's the newest technology we're trying. The fancy term for it's called spatial transcriptomics. But in practice, what we're really doing is we're looking at a specimen under the microscope, and we're also able to sample DNA and barcode it based on where in the image it was. So if I look at a specimen and I see, here's like a little region up in the top right corner that has like a whole bunch of like parenchyma cells in a funny shape. I'm like, what's going on over there? I can then look at the barcodes that correlate with that region. Or I could just stain it directly and look directly at the genes in that region and see what genes does that uniquely have expressed that I can then identify those cells.
Right. So you're looking at the cellular level of microscopy. I was wondering whether you were down at the actual protein level.
So it depends. There's two different methods. One is called in situ, where you're putting it inside the physical specimen, and that relies on a number of barcoded probes. So you take a bunch of RNA of known sequence, and then you can add any barcode you want to the end of the RNA. And then you have that RNA go inside your specimen and bind to its complementary sequence, and that's called a probe. And then it'll have some barcode on it that'll give it a fluorescence under a certain circumstance, so that you can then piece together which one's which. And then based on the number of letters you add on, you can have combinatorial expansion. So you get to like 100, 1,000, 10,000 species of RNA you can put in at once. And then all you do is you stain the membrane, so you can tell which cell is which. And so you end up with an image where you see a cell full of tens of thousands of little dots. Ideally, right now it's about 350, I think is the best I got, but they're kind of trying to push towards that larger number. So you come up with a list of 350 RNA molecules that you want to ask about, and you create your custom probes. You put your probes on there, and then you image it, and you take a series of images where, like, you know, round one has a signal, and then round two doesn't, and then round three doesn't, and then round four has a signal, and so on and so forth. And then you stitch that together into a barcode that tells you, oh, this species had these 4 lights across this many signaling opportunities. The specifics isn't important. What is important is that when you're done, you see an image that has 350 different colored dots in it, and that you can count the dots and say how many of a particular gene is in this particular cell. And so you're seeing it at the subcellular resolution, counting actual RNA molecules directly.
I was wondering, and you reminded me, because many years ago, we were able to create some fluorescent mice, I believe, in a similar sort of way. But I was wondering whether, because about, I don't know, 50 years ago, protein synthesis was worked out, except that we didn't know how the final polypeptide chain managed to wrap itself up into the complete shaped protein. But then, much more recently, there's been a computer program which can do that. So I was wondering whether you were trying to look at both ends of that process. You know, here's the gene sequence. Here's what the computer has worked out would be the shape of the protein, the tertiary structure of the protein that's derived from that particular gene sequence. I was wondering whether you were trying to look under, this would have to be a very high-powered microscope, possibly we're looking at electron microscopy here, to see whether you could actually see that shape.
So the tool you would need to do that is called Xray crystallography. So you would get your, you would have your synthesized protein. You would take they do the fold at home or the crowdsourced folding games, and they'd figure out the best conformation to fold. And then you would try to, I don't know the actual means by which you would control the folding, because I'm not a synthetic biologist, but if you wanted to then see exactly what it looked like, you would then have to crystallize the protein by making an actual crystal structure where the protein's repeated in the same orientation. Then you shine X-rays on it and then mathematically look at how the X-rays bounce off. Yeah,
so then we're going right back to the early days of working out the actual shape of the DNA molecule itself.
Yeah, that's like Rosalind Franklin level of
who never got the recognition she deserved.
Yes. That's you know, academia for you. The guy who yells loud enough gets all the credit.
And sadly, she died rather young of the very cancers that we're probably capable of helping solve now.
I'm sure she was following all the OSHA guidelines for X-ray exposure during her work. Like,
well, I mean, yes.
There's plenty of horror stories that have made me quite paranoid.
Yes, they're going right back to Marie Curie.
Yeah. There was that one chemist in the 90s who like dripped like, I think it was like ethyl mercury on her latex glove, and it like went through her glove into her hand, and she just died slowly over the next year of mercury poisoning, and nobody knew why. Like, so I mean, fortunately we have much better safety standards now, but there's some pretty dangerous stuff you can work with.
Yeah. Again, don't try this at home.
No, don't. Don't pour mercury on your latex gloves.
No. Fortunately, ethyl mercury is not very accessible. So, although they had was it Maramata, the bay in Japan, that had their outbreak of mad human disease that was through eating too much tuna, which, I don't know if you know this story. there was a factory in a bay in Japan, which had an outflow that contained quite a lot of organomercury compounds. And so the bay was polluted with this rather nasty poison, went up through the food chain. The top predator was a tuna-like fish, which they caught and ate. And the cats were fed the bits that the heads and the fins, the bits that humans didn't like to eat. And after a while, they noticed that the cats were walking about like drunks and falling over and dying. And then it started to happen to the humans. And that was really how we first discovered that mercury was a serious poison. And up until then, we've been playing with it, sticking our fingers in it, floating coins on it and everything. Yeah. Anyway, enough of that nonsense. The future, where are you going to go with this?
Are you asking about the spatial technology, or? Yeah,
you and this research. How soon is this going to be really beneficial? Rolled out.
I mean, it's already being offered for there are companies called contract research organizations that you can now create basically a virtual pharmaceutical company if you have any money on you. So like a lot of investment firms will just throw a bunch of money at what's basically like an embryonic incubator startup company that has zero lab. And then all you do is you just order, like you go to like a tissue bank and you order, like, I want some biopsy samples from like this type of tumor. And then you have them sent to a CRO lab and then they process it. Those CRO labs already have it up and running. And so you could then start asking questions like, why are the immune cells not attacking this tumor specifically? Where are they? What's different about the different regions that they're in? How are they clustering with each other? What are they saying to each other when they cluster with each other? And then this information could be used to create a very nuanced approach to help augment that checkpoint blockade I was telling you about. So we know that they're exhausted, but we don't know how specifically they are in this tumor and are there common elements we could find in there. Before, you couldn't quite tease it out. You'd have to sort of infer it. You'd look at it under the microscope, you'd see some regions, you'd sequence it, you could even pull it apart to a single cell level and do single cell sequencing. You could see some signature genes, but you didn't know which genes were where, and you didn't know who they were talking to. And now you can do it all at once. So I think that's really the biggest way it's going to move the needle and give very nuanced views of things like autoimmunity or immune oncology.
Amazing. Roll on. More power to your elbow.
Yes.
And how is your career going to develop? Have you got any? Can you see into the future in that?
So I mean, I was recently kind of made one of the co-leaders of our entire facility. So I'm helping to integrate all of the different methods into a single menu. So by default, our facility will do traditional like whole RNA sequencing. So somebody has some experiment where they, generate a whole bunch of either cells in a petri dish or mice in a lab mouse experiment. And they want to ask like which sets of genes are different between them. We sequence them right now, but we also want to give them the option to do single cell sequencing or spatial transcriptomics so they can create these more holistic views. And we want to link that to other readouts like proteins or cell readouts so they can create this kind of general Google Earth omics view of everything using standardized experiments so that we can kind of compare things to each other. In the past, people would just do these little ad hoc experiments where they would ask a very specific question about a very specific context, but it wasn't generalizable to anything else. Now we're trying to make them more standardized so that we don't keep reinventing the wheel or keep redoing the same experiment over and over again.
Yes. Yes, a standardized procedure would be much more useful. Yeah.
Yes.
So, moving on to lighter things, you enjoy all forms of speculative fiction.
Yes.
So, okay, Star Wars or Star Trek?
Yes.
Now you got to choose.
Oh, I have to choose? That's tough. Like, I don't know if I could choose. It honestly depends on like what mood I'm in at any given point. I suppose I liked Star Wars first, but I've enjoyed Star Trek more as an adult. So I don't know that I could choose. I know some people get very tribalistic about it, and they're like, "Oh, only one or the other." But
Yeah. Gene Roddenberry was well ahead of his time, wasn't he?
he was. I think his work in getting it up and running with him and Lucille Ball getting like the original Star Trek up and running was groundbreaking. But I think like there's a trope called growing the beard, which is the opposite of jumping the shark. So I don't know if you've heard of those terms.
Nope.
So have you heard of? Are you familiar with the show Happy Days and the Fonz? And
Yes.
yeah, so Happy Days kind of kept running and running and running, and eventually the writers just got kind of burned out or bored, and the episodes are getting more ridiculous until eventually there's an episode where the Fonz did like some ridiculous stunt where he did like a water ski jump over a shark, and everyone's like, all right, this show has been on way too long. It's it needs to stop.
Yes.
But the opposite of that is called growing the beard. And that's in Star Trek: The Next Generation, where in season 3, Riker grew a beard, and the show really became amazing from season 3 onward. Like it was good, but it was a little stuttery for the first 2 seasons. And ironically, it was because Roddenberry died at that time, and the writers really were able to fully, realize their potential as the writing team for that. And then Star Trek of the 90s became what everyone really was obsessed with for then. So I think like it's that sort of founder's principle. The founder has a great initial idea, but sometimes you have to let the experts do their thing.
Yeah. Once you've got a good team, you should step back a bit. But the interesting thing about these fiction scenarios is because it's not on Earth, you can do things that are culturally not, that they're too far off, too far ahead, that we wouldn't be able to accept if it was done, with a background of reality. But since it's on board a spaceship, then you can do the first interracial kiss or the first homosexual hug or something, because it's that removed from reality. Think of that.
I think in principle that seems like it should be the case, but I obviously wasn't alive when the first interracial kiss happened or what the reaction was. I can't imagine there were people on the religious right who took that sitting down. And even today you see people on the internet just going crazy about are complaining about SJW wokeism contaminating their Star Trek when they have too many gay or trans characters show up. So I think you're always going to find bigots who have their complaints about anyone other than just boring white males doing everything in the show. But I think what speculative fiction allows for is the democratization of the thought experiment, by allowing you to ask questions like the Star Trek thought experiment of like, if you go on the transporter and it breaks you down and reassembles you somewhere else, are you the same person? Or if your mind gets uploaded to a computer and copied and something happens to your original, like which one's you? And those types of more outlandish scenarios that can really force the philosophical examination of identity. Black Mirror, I guess, is the more like popular recent example of that. But
Of course, they only use the transporter as a cheap way of getting from place A to place B without having to film some sort of journey.
Of course, and yet, like, it raised that very question. Now, I believe canonically, like, you still have a continuity of consciousness, but there have been other like fictions that have really explored that more deeply. There was a video game called Soma, which is very well done. That was sort of like a psychological horror game that's based around the idea of like, what if when you upload your like say I scan your brain and I create a simulation of what your brain is doing right now.
Yeah.
Is that simulation merely a description, or does that simulation itself experience things? And they're saying, well, what if it did experience things? Let's assume it does, and let's like explore those implications. If I copy it, if like, let's say you need to copy yourself in another body, and so now you wake up and you're in the new body, but your old body was just there. But your old body is going to be like left to some horrible fate. Do you kill the body that was just you a second ago? Like, is that even you? Like, or are you just a new being who came into existence a second ago and now is murdering the other self? That's where it gets really, really like confusing. And
Yeah.
horrifying in some ways.
We'll be touching on something like, for example, the ridiculous idea that a son can be the same as his father. Where have I heard that?
I mean, that's more. I honestly can't square that circle. Like, and I'm told that my inability to do so is merely my ignorance of the complex theological principles, which very may well be the case, but that doesn't change the fact that I can't square the circle. And unless and until I can't swallow that pill.
Incidentally, the Pope recently came out as being against the cancel culture and deplatforming. So maybe we've reached the end of that woke period. If he's, saying things like that.
So the Pope is now opposed to it. I think cancel culture is also a bit of a silly way of framing what the market has been doing the whole time. I mean, people have been trying to cancel The Simpsons all of the 90s for every which little offense. You've got Tipper Gore trying to cancel violent video games. You've had, everyone's like, there's some media out there that somebody doesn't want to exist, and they'll complain about it on whatever platform they can.
Yes, but out of all the people who you might expect to be biased or bigoted, for the Pope to come out as saying that he's against this sort of justice by media is quite amazing.
Yeah, I have no idea where he's coming from with that.
He also said that if you don't have kids, you're selfish, without revealing whether he's got kids himself.
Well, I don't think he's supposed to have kids, but the irony of that is, man who's ostensibly a virgin complains about people who don't have kids.
Yeah.
I mean, the Catholic Church has always been about almost not quite quiverful levels, but the whole 6 kids Catholic joke that Jim Gaffigan makes. That's pretty consistent with their doctrine.
Well, there you go. Do you know, I think we've almost come to the end of our Freethought hour, and it's been interesting. You've been very easy to talk to. You've been very informative, and I thank you very much for coming on and gracing me with your presence. Sorry about my phone going ping. I keep forgetting to turn it onto airplane mode. So I'm going to release you now and suggest that you crack open a bottle of beer like I've done. and thank you very much. And thank you, everybody. Thank you, everybody,
for the invite.
Thank you, everybody, for watching. And there will be another Freethought hour next week. I'm still negotiating with potential guests, so I can't tell you who it's going to be, but we will have somebody as wonderful as Seth, hopefully. So, Seth, thank you very much.
Thank you.
Take the rest of the day off.
Will do.
Bye-bye.